However, his field of work is not exactly gynecology and obstetrics, but andrology. His area of investigation was dedicated and is dedicated to sperm physiology and andrology, and has even a patent approved by the FDA of the United States. Currently, he is the director of Androgen. Androgen, here near us in Coruña, is the andrology clinic. It's a very famous andrology clinic in Spain. I asked Alberto Barros to tell me personal skills not linked to the clinical part and the research part. And he told me that you are a musician
Yes,
and a composer.
Yes, I am.
And including you have performed or produced, I don't know, a Portuguese fado.
Yes, well, no, it's a Galician fado.
Finally, and to end my presentation of it, I want to say that Professor Juan Alvarez is a very close friend of many Portuguese colleagues, also, of course, of the Portuguese people. Thank you very much, Juan Alvarez, to be our Palestinians. Thank you very much.
It is a pleasure. Thank you very much for the introduction, Mario. I have to say that I have two grandfathers that are from Portugal. So, you know, I have many roots in Portugal. As you said, you know, my main interest is in the field of andrology, but my research was actually focused on oxygen radical chemistry and biochemistry and its implications, you know, in sperm viability and sperm function. And from that point is where I became interested in DNA damage, because in 1991, Bruce Ames, which was a very prominent scientist from the United States, from Berkeley, he was measuring oxidative damage to DNA sperm and was measuring oxoanosine, you know, in urine and in semen. And at that point, which was in 1991, is when I became interested in DNA, you know, damage. And I got connected with Donald Evanson, who is the one that developed the SCSA assay. It was the first, you know, sperm DNA test developed originally. Then after that, you know, it came the tunnel, it came the allosperm, it came the comet, you know, many different tests. But I'm also a PhD in physical chemistry. Don't forget that. That's actually my passion, is physical chemistry. It's not precisely, you know, being an MD or a biologist, but everything, you know, can be combined. So you can become, you know, you can go farther inside, you know, the cells and the biology, and actually is very helpful. And I also have a PhD in reproductive biology, okay? And remember that PhD doesn't mean that I'm a doctor in philosophy, okay? Somebody introduced me in a talk, and he said, Dr. Alvarez, besides being an MD, he's also a doctor in philosophy. And I said, no, PhD, philosophy doctor, doesn't mean that you're a doctor in philosophy. It means that you are actually have a doctorate, you know, in literature or chemistry or whatever it is. Anyway, let's start with the talk. As you can see in the title, can you hear me well? Okay. In the title, it says clinical implications of sperm DNA damage in assisted reproductive technologies. And I'm going to start with, let's see, I'm clicking here. With this paper that was published in 2010, which is an invited review from Fertility and Sterility that I published with Dennis Sakas, which he's very well known in the field of sperm DNA fragmentation. And at the time, he was a professor at Yale University. And we were at a meeting in Barcelona, the ESHRE meeting. And after he gave the talk at the meeting, you know, I met with him and I told Denny, you know, we have to write a review about sperm DNA fragmentation, because after, you know, I'm talking about since 1991 to 2008, there were many publications about sperm DNA fragmentation, and there was not any evidence that those tests were measuring, what we would consider, critical DNA damage that will have an impact on pregnancy outcome or embryo development. So this paper that you see here is creativity. You were saying before, Mario, that I'm a musician. The same thing applies to writing a paper or, to do science. You have to have a creative mind, and develop ideas. So that's the way this paper, came about. So 90% of the paper, although there is some data about the use of testicular sperm in couples with repeated IVF failure, is just a paper of thinking, what can be done or what could be done at the time, to improve our insight into the sperm DNA damage. So anyway, the main conclusions of this paper were these two, that we have repairable sperm DNA damage and non-repairable DNA damage. Like, for example, single-stranded DNA breaks can be easily repaired by oocytes from young women or egg donors, or even, older women. But there is some non-repairable DNA damage. That is the one that is going to have consequences on pregnancy outcome and embryo development. And this is the one that we should measure. But at the time we wrote the review, there was no test that could measure that kind of damage. We just measure, a combination of single and double-stranded DNA damage. And the second conclusion of the paper is that most sperm DNA damage is produced at the post-testicular level. It's not produced in the testes. Actually, there are many papers published where they show, using the tunnel test, for example, that, as you will see in the next slides, sperm DNA damage in testicular sperm is very low compared to ejaculated sperm. So let me show you. First, when we talk about repairable versus non-repairable, that's just what I said before. At the time, we wrote the paper. At the time, not now. We didn't have a test that would measure non-repairable versus repairable DNA damage. But recently, and I'm talking about maybe a couple of years, a highly sensitive test that measures double-strand DNA breaks in the sperm DNA has been developed, which is the common fertility at neutral pH. At neutral pH, you measure double-strand DNA breaks. And also that uses artificial intelligence software, because when you do a test, it has to be accurate and precise as well. And this is the one that we are using right now at our center and the clinics in the US that I work with. I collaborate with clinics in the US with gestational surrogacy cycles. And the other thing is that double-strand DNA breaks, or DSB, when the value is above 60%, is associated with pre-implantation block, day 3 embryo block. 60% means that out of 10 embryos, only 4 reach the blastocyst stage. The other ones are blocked because, as you well know, the paternal genome starts being expressed after day 3. And if there are double-strand DNA breaks that were not repaired, the embryo will not have the information required to develop. So that's what 60% means anyway. But also, you can have maybe a normal embryo development because those DNA double-strand breaks are at a level that is beyond embryo development. It's after implantation. What I mean embryo development, the initial embryo development until day 6. But it can also affect sequences that will have repercussions in embryo development after implantation. So you will get maybe a recurrent miscarriage, or you can get just pregnancy, or implantation failure. So this is the main consequences. As I show you here, you can consider the sperm DNA, as a manual of instructions. Maybe at the beginning of the manual is where you have the instructions that if they are affected, it will interfere with embryo development, up to day 6. But if the pages that are affected, are towards, the end of the manual, you will have implantation failure at week 6, 8, 10, or even 12. So that will be the difference between pre-implantation block and post-implantation block. Now, it has been studied repeatedly, and has been shown that most of sperm DNA damage is produced in the epididymis. And you will see later on the tables that I'm going to show you. This one, for example, you can see here, in these patients, from 1 to 12, that the sperm DNA damage measured by the tunnel test that measures both single and double-stranded DNA damage is much higher than testicular sperm. Okay, you can see here, the average is 13.3 versus, or 40 versus 13. So we know that most of the damage has to be produced at the post-testicular level, and that, in the testes, maybe this patient has a pathology that increases, a little bit more sperm DNA damage in testicular sperm, like for example, varicocele. But as you can see, in most of the cases, the DNA damage is much lower in the testicular sperm versus the ejaculated sperm. Okay, so is post-testicular DNA damage associated with single or double-strand DNA breaks? When we wrote the paper, and until recently, and actually Mario is involved with me in this study, we thought that most of the damage produced post-testicularly, it was single-stranded DNA breaks, because oxygen radicals cannot impact simultaneously, in both strands at the same points. So it makes more sense that, oxygen radicals by themselves, which, as you know, are very abundant in the epididymis, especially in the coda epididymis, that will induce single and not double-strand DNA damage. And so if the single-stranded DNA damage, like SSB, can be easily repaired by the oocyte polymerases, that would not explain the poor pregnancy outcome, that people claim when a sperm DNA is damaged. On the other hand, if the post-testicular damage were to be associated with double-strand breaks, DSB, that would explain the poor pregnancy outcome, especially when that type of damage cannot be repaired, which is in most cases, although not always. There are cases with oocyte donors, young oocyte donors. They can have oocytes that can repair double-strand breaks, but also depending where they are located. Now, this is something new, and that we are going to publish this month in the Journal of Reproductive Assisted Reproduction Genetics, where there is enough evidence for several groups that mature sperm has endonucleases in the nucleus, just behind the nucleus, and that if they are activated, they can produce double-strand DNA breaks. Now, if that were the case, this is a hypothesis that we are actually postulating, but if that were the case, that would explain why when you use testicular sperm in couples with repeated IVF failure of day 3 embryo block, why the pregnancy outcome improves significantly. I mean, you go from 0 to 50%. That's quite significant. Now, where are double-strand DNA breaks generated? Because 2 years ago, with the scientists that actually found that the ZIMOT device, or also called fertile chip, I don't know if you are familiar with that, but it's a device that reduces double-strand DNA breaks by 50%, okay? So it has an inlet, port inlet, where you put the sample, the semen sample, and the other side, you retrieve at the other port, the outlet, you retrieve motile sperm with a reduction of 50% in double-strand DNA breaks. So when I spoke with this scientist, he told me that most of the DNA damage was produced in the testes during spermiogenesis. Remember, when the remodeling of the chromatin, you have topoisomerases with ligases, and also, with cutting DNA double strands, as both activities. And I asked him, and I said, Agustin, how can you explain, if that were the case, that most of the damage comes from the testes, that when we use testicular sperm, pregnancy rates improve so much? So he said, well, I don't know. I don't have an explanation for that. I mean, Mario's group, led by Dr. Alberto Barros in Porto, in the Center for Genetics of Reproductive and the urologist Luis Ferraz, they have been doing this for how many years, Mario? For how many years have you been doing TESA-ICSI cycles? Can you hear me?
Yes,
you can activate your microphone. Well, okay, it doesn't matter. They have been doing it for more than 20 years, and they have excellent results, when they use testicular sperm. So that will not go in line with what
I forgot to take out the sound. What was the question?
For how many years have you been doing the TESA-ICSI cycles in repeated IVF failure patients?
How many cycles?
No, how many years?
With Alberto here?
Yeah.
After France, it was 1996.
That's what I thought, yeah, more or less.
1996.
And you do it because you have excellent results, although you don't
Yes, of
have
course.
any scientific evidence of why it works. Anyway, so I told this scientist that developed the Zimo device, which a very famous geneticist here in Spain, and he didn't have an explanation. Well, now we think we have an explanation, and it's because most of the DNA damage, double-strand DNA damage, is produced in the epididymis, not in the testes. The one produced in the testes is perhaps 10%. So there you have double-strand DNA breaks can be generated during spermiogenesis with the double activity of the topoisomerases that cut and ligate the DNA to accommodate, the DNA with the protamines. So this is one of the main mechanisms during, spermiogenesis. This type of damage can be apparently prevented, at least in part, with xanthines, like avoiding xanthines like the coffee, tea, etc., that are known to inhibit topoisomerases. Now, at the post-testicular level, due to the activation of sperm nuclear endonucleases and during passage through the epididymis, also can be produced, and I think, or we think, is the main source of double-strand DNA breaks, and in this case will be induced by oxygen radicals. Now, there is a third that I'm going to try to show you if I manage to, let's see. Barbara, how do I go back to my desktop?
So you should stop sharing the screen.
Okay.
And try to find the other document. Okay.
Well, let me continue. I don't think it's going to work. I'll try to do it at the end. Okay. I will show you a couple of tables where you can see that, where it says iatrogenic DSB damage after incubation post-thawing. That means that when you incubate the sperm after thawing, there is a burst in oxygen radicals during thawing. And we have already evidence that after 20 minutes of incubation at 37 degrees, that there is an increase in double-strand DNA breaks, which is something that I constantly tell, embryologists, in many different labs, especially in the United States, because I work with 15 different clinics in the States, that after you thaw the sperm, you should just microinject the oocytes immediately. You should not be waiting for much longer, because you might induce iatrogenic double-strand DNA breaks. Now, it happens that we develop a new method of sperm, not new method, the only one, of sperm vitrification in straw. It's called the Vitri straw technology. And this is being commercialized now in Spain, and will be also commercializing it in the United States. But what I wanted to tell you is that vitrified sperm, when you devitrify the sperm, they don't produce oxygen radicals. They don't produce this burst in oxygen radicals. So there is not an increase in double-strand breaks. They stay the same for an hour. However, when you use slow freezing, which is the one that we are using currently, the percentage of double-strand DNA breaks can increase by 2 or 3-fold. And it increases as a function of time. So that's what I wanted to show you with those tables. But since we might have some problems going in, back and forth, I just will continue. Should these results be confirmed, in human sperm, you will explain why testicular sperm improves pregnancy outcome and prevents day 3 embryo block. Now, the only way to prove that this is the case, that double-strand DNA breaks are much lower in testicular sperm than in the ejaculate, is to take a biopsy or a TESA sample from the testicle and retrieve the sperm one by one, like when you do ICSI. You take it from the microdroplet and analyze them one by one to do the comet at neutral pH, measure double-strand DNA breaks, and then compare that with ejaculated sperm after doing, a swim up or a gradient. Therefore, the comet, test or the comet fertility test, a neutral pH could be of great clinical utility in the diagnosis of couples with day 3 embryo block or repeated IVF failure. In fact, all the patients that I saw to date with day 3 embryo block, they all have levels of DSB, double-strand DNA breaks, of above 70–80%. So that obviously explains why you have day 3 embryo block. All of them, without exception, all the couples with day 3 embryo block, the oocyte is rarely involved in day 3 embryo block. It's the paternal genome. So I think this is very relevant that you have a test like the comet fertility that I just mentioned before to measure double-strand DNA breaks, because now we have a device that can lower that value by 50%. That has been published. Don't remember the journal. I don't know if it was Fertility and Sterility, but you should look for that paper. And the other option that you have is to use testicular sperm. Obviously, many males will, and also embryologists, don't like that option, I don't know why the embryologists are very nervous, when they use testicular sperm. But this device that I mentioned, the ZIMO device or fertile chip, cannot be applied to semen samples that have less than 5 million sperm, in total, motile sperm. So in those couples with repeated IVF failure and with less than 5 million motile sperm in the sample, you cannot use the fertile chip or the ZIMOT device. So you have to use testicular sperm. And I can tell you that my experience, Mario's experience, Dr. Barro's experience, Dr. Luis Ferraz' experience, Dr. Ferran Garcia in Barcelona also has a great experience, like going in couples with repeated IVF failure to pregnancy rates of 50%, and you have an idea why that might be the reason. Well, and this is, our webpage. It is always a commercial, after every talk.
Thank you very much for being with us, Juan Alvarez, and I hope we'll see in the future. Barbara,
Okay.
you can.
Thank you for inviting
Sandra,
me. Okay.
Thank you very much.
Thank
Bye,
you.
bye.
Thank
Bye.
you.
Thank you.
Bye. Thank you.